Welcome to EPG Pathshala. I am here to discuss about structural genomics. Beginning with learning outcomes, after studying this module, you shall be able to understand the concept of structural genomics and appreciate the need of it. And you shall also be able to understand the goals of structural genomics study. You shall be able to decipher the steps involved in the process of structural genomics, and also you will be able to describe the various techniques like NMR, X-ray crystallography and TROSY. You shall be able to identify the role of structural genomics in varied fields of biological sciences, and you shall also be able to describe the future prospects of structural genomics. You shall be able to understand the outcome of this structural genomic studies as well. Unified understanding of molecular biology from atomic to cellular level come from investments and efforts in two fields, namely structural genomics and functional genomics. Structural genomics deals with the large scale determination of three-dimensional protein structure, whereas functional genomics deals with the characterization of the biological function of biomolecules. Janet Newman of Structural Genomics, San Diego, USA, describes structural genomics as any program of structural determination that uses genomic sequence data available as its driving force. Advancement in technologies combined with the recent flood of genomic sequence data laid a basis for the worldwide effort to determine the structure of proteins. Protein structural determination generates valuable clues for predicting the protein function, understanding biochemical functions and also the evolutionary relationships. Many workshops and meetings were organized and sponsored by both the public and the private funding agencies in the United States, Europe and Japan on what could be achieved through this venture of structural genomics and how the international collaboration could help. US funded efforts aimed at broad coverage of protein structure space, technology that would be required for increasing both the speed and scale of the protein structural determination, ranging from gene sequence to disseminated structures. Private sector efforts and funds are basically focused on those proteins which represents their drugs or on the proteins which are medically relevant. Number of structural genomic centers were started around the world to solve the protein structures using both the X-ray crystallography and NMR. This table in this slide is basically representing the various centers where the structural genomic studies are being carried on, and along with it the leaders, their website and also the topic on which the structural genomic studies are being relied on. This is all represented in this table. 5 basic goals or objectives of structural genomics have been defined. These are laid here. First is identification of best high throughput technologies and the most efficient processes to convert gene sequence into three-dimensional structure information. The second important goal of structural genomic study is the determination of protein structure, their functions, evolutionary relationships and also the molecular mechanism. Third goal includes the creation of a complete repertoire of protein folds and experimental macromolecular structures. The fourth goal of structural genomic study includes the study of biomolecules which are highly informative, such as those obtained from the model organisms and also with the medical relevance. Fifth important goal of structural genomics is to obtain structural information within 16 to 12-month time period from cDNA and also reduction in the cost of each structure by 90%. The first goal, that is the identification of the best high throughput technologies and the most efficient processes to convert gene sequences into three-dimensional structure information, requires two techniques, namely X-ray crystallography and NMR spectroscopy. NMR stands for nuclear magnetic resonance spectroscopy, that provides the majority of experimental data for three-dimensional structural determination. Currently, X-ray crystallography seems to be the probable workhorse for structural genomics because if provided with a well-diffracting crystal, it is quite possible to determine three-dimensional structures in hours. However, the throughput of structural determination using X-ray crystallography remains vague because the rate determining step continues to be the production of well diffracting crystals, a process which is quite unpredictable and can take between hours and months. On the other hand, the structural determination of proteins by NMR studies does not require any crystals, and structures can be determined within minutes of the protein being purified. Currently, NMR is also limited because of many reasons, and the reasons include size constraint of samples, then lengthy data collection and analysis time, which often requires a period of month, and this method basically applies to proteins which are smaller than 250 amino acid. So, because of this point, NMR is limited currently. Second important goal deals with determination of protein structure, their functions, evolutionary relationships, and also the molecular mechanism. Protein structure represents a powerful means of discovering function because structure is well conserved over evolutionary time scale, and therefore, it provides the opportunity to recognize homology which is undetectable by sequence comparison. The literature also suggests homology inferred from the structure. For example, there was an unexpected similarity observed between actin and 70 kilodalton heat shock cognate protein, top rim domain shared between some topoisomerases, primases and nucleases enzyme, and also the highly similar constant and variable domains of immunoglobulins. Indeed, most evolutionary relationship cannot be inferred from sequence. In addition to this, three-dimensional structure of a protein can also yield direct insight into its molecular mechanism. For example, structure of Tata box binding protein, in short TBP, when bound to DNA, not only provides the clue of how this molecule is interacting, it also gives us a sense of DNA binding specificity. The third important goal of structural genomic study is the creation of a complete repertoire of protein folds and experimental macromolecular structures. The creation of this complete repertoire of protein folds requires surveying the structure of proteins of different families, studying the individual protein domains, experimentally solving the structure of one representative domain from each family, which will be augmented by computational comparative modeling so as to provide the coordinates and model structures for related proteins. The fourth important goal of structural genomics is the study of biomolecules that are highly informative, such as those from model organisms and with medical importance. Many structural genomic projects run the gamut from exhaustive studies of all the proteins found in model organisms such as Methanococcus janaceae, Mycobacterium tuberculosis, Mycoplasma genitalium, Mycoplasma pneumoniae, Escherichia coli, etc., and of relevant medical value. This include, for example, investigating the proteins involved in disease processes such as protein kinases, oxidoreductases, phosphatases, and other metabolic enzymes, as well as signal transduction proteins. The last and not the least important goal of structural genomics include to obtain structural information within 6 to 12-month time period from cDNA and reduction in the cost of each structure by 90%. Now, coming to steps in structural genomic study, the process of structural genomics includes first the selection of the target protein, then followed by that the PCR amplification of the coding sequence from genomic or cDNA, cloning of the selected target sequences frequently combined with the recombinant base vectors that will allow the creation of many different constructs. This vector will incorporate different affinity tags such as his tags and glutathione S transferase to aid in the process of purification, as well as promoters that will allow trials in different expression systems. Steps in structural genomic studies. So, following steps are being undertaken in structural determination of proteins starting from gene sequence to disseminated structures. The process basically includes first selection of the target protein, second important point is PCR amplification of the coding sequence from genomic or cDNA, third cloning of the selected target sequences frequently with recombination based vectors that allow the creation of many different constructs. This vector incorporates different affinity tags such as his tags, histidine tags, glutathione transferase GST to aid in the process of purification as well as promoters that allow trials in different expression systems. Expressing the protein at a sufficiently higher level is the next step in the process of structural genomics. Then sixth step is basically sequencing the clone gene to verify that the coding sequence was correctly amplified and confirming the identity of the expressed protein and characterizing it as a precursor to NMR or crystallographic studies. Seventh important point is obtaining the protein in sufficient amounts and purity for either approach, and then next step is defining suitable crystallization or NMR solution conditions, NMR or X-ray crystallographic measurements, and also determining and refining the experimental structure. The next step includes calculating comparative protein structure models using this new template, and last step is making functional inferences from the structure plus derived models and disseminating the findings. This flowchart represents the processes that are involved in high throughput structural genomics using X-ray crystallography. So, N over here indicates that the process has failed and Y represents that the process has succeeded. So, basically this process begins with choosing the target, then cloning the coding sequences of it, and then followed by its expression either as soluble protein or as a insoluble protein. If soluble protein, then it is sent to the next stage of purification, and for quality assurance and biophysical analysis, this previous step will tell whether the protein is crystallizable or not. If the protein is crystallizable, then it is sent to crystallization trials and microcrystals will be formed. This microcrystals will be showing the diffraction pattern. So, they will be called as diffraction quality crystals. They might be containing methionine or they might not contain methionine. If they do contain methionine, we will be obtaining selenomethionine crystals, and the MAD data, which stands for multiple anomalous diffraction data, will be collected, and this data will then be allowed to go to next level, which is phasing, model building, refinement. So, finally, the structures will be deposited in PDB, which is a database of the protein structures, or otherwise it will be used as to form the models, homology models, or annotating these structures. If the protein is unlikely to crystallize, then the next step would be to identify and correct the problem by subcloning it or by adding metal or cofactors. If still the protein does not crystallize, then that is sent for NMR studies. NMR stands for nuclear magnetic resonance study. So, this data obtained from NMR is then again deposited into the PDB to create homology models as well as to annotate structure. And if this protein is able to crystallize itself, then some of time it might not contain methionine. So, in that case the data will be sent, or this protein structure will be sent for MIR analysis, which is multiple isomorphs replacement analysis. The MIR data will be collected and again it will be sent for phasing, model building and refinement, and finally depositing the structures in PDB, which is a database of protein structures. So, and creating homology models and annotating structure. Coming to challenges in structural genomic study, the experimental structural genomics faces many bottlenecks to overcome. Moreover, many individual proteins are expected to be intractable without extensive effort. Therefore, parallel studies on related proteins are being relied on to increase the likelihood of readily solving a structure for a family of proteins. The success progress rate of individual protein targets through experimental process will be like a kin funnel, with many targets starting at the same time and a fraction failing at each stage of the process. So, for this thing, please refer this table 1, which is basically representing the status on one column, the total number of targets starting at, in another column, percent success rate, in third column, and overall percent success. Technology forms a foundation for structural genomics. Structural genomics is thought to be a technology driven field. Technology development is important for the success of high throughput structural determination. Key foundation for structural genomics include development in various fields like macromolecular X-ray crystallography, NMR, high throughput technologies, general system for cloning and expression of proteins. In X-ray crystallography, technological advancement aid in determining the protein structures efficiently within ease, and the technological advancement includes the use of synchrotron radiation and its tunability in collecting multiple anomalous diffraction data on protein crystals for mad phasing, the use of selenomethionine in crystallographic phase determination, third, automation of nanodroplet crystallization, cryocooling techniques that can reduce radiation damage to the crystal, and the automation of the structural determination process for macromolecular crystallography. NMR, nuclear magnetic resonance technique, also sees many advances, which includes availability of cryoprobe high field NMR devices, development of multidimensional techniques for data acquisition, introduction of automated procedures for data analysis, and new techniques such as transverse relaxation optimized spectroscopy, it is also known as TROSY, suggesting that NMR structure determination will head towards even higher throughput. High throughput technologies have been developed in order to increase the structure determination throughput rate. Automation, miniaturization and parallelization of the process steps can also deliver increasingly higher rate of protein structure determinations. New technologies such as cell-free protein production and nano volume crystallization can facilitate high throughput preparation of protein samples. Expressing high levels of soluble protein is a particular challenge. So, there is a considerable interest in fusion between the target protein and green fluorescent protein GFP that fluoresces only when soluble and folded, therefore, indicating the folded proteins in solution. Cell-free expression system hold great promise for improving the yields as well as allowing the production of toxic proteins. Another optimization is the use of hypothermophilic proteins, which are easier to purify when expressed in mesophilic hosts, as these proteins are resistant to heat that will denature most of the proteins of the host. This slide basically represents the flowchart, which is depicting the utility of data obtained from structural genomic studies in varied areas of biological science. Data from structural genomics serves as an inoculum for scientific discovery in the varied areas of biological science, which include biodiversity, molecular genetics, molecular biology, molecular medicine, biotechnology, breeding of farm and domestic animals, functional genomics, toxicology, agriculture plant genetics, molecular evolution, pharmacokinetics, human health and diseases, immunology, bioinformatics, molecular biophysics, proteomics and biochemistry. Coming to structural genomics and molecular medicine. Structural genomics aid in development of various molecular medicine programs. Some examples are enlisted here, which will represent that how this structural genomics is helpful in the development of various medicines. Some example here includes, first, the peroxisome proliferator activated receptor gamma, which is also called as PPAR gamma. This is a transcription factor involved in the regulation of adipocyte differentiation. PPAR gamma agonist basically enhances insulin sensitivity. Some are in clinical use against type 2 diabetes. So, knowledge of this structure of this receptor will help in designing novel structurally dissimilar agonists that might be as effective as and less toxic than the agents used now in the clinics. Structural determination of RNA polymerase from hepatitis C virus will also help to design highly specific inhibitors of this enzyme. Then third point, the potent tetanus neurotoxin from Clostridium tetani binds to gangliosides on the surface of neurons in order to enter the cells. Once it reaches the CNS, central nervous system, it interrupts the neurotransmitter action, causing spastic paralysis. So, knowing the structure of this toxin bound to the carbohydrate part of the gangliosides, it will become easy to design inhibitors of this toxin and model the interaction of the similar botulinum toxin. Structural genomics as a functional genomics tool. So, structural genomics provide unique insight into the protein function. And key idea behind this is that the protein structure is better conserved than the sequence. Example below illustrate how it acts as a valuable tool in elucidation of the protein function over and above that obtained from sequence alignments, expression analysis and currently available homology modeling algorithms. The TUB gene, which is responsible for obesity phenotype in tubby mouse strain, is a transcriptional regulator. The three-dimensional structure of the core domain of murine tubby revealed the presence of a positively charged group suggesting a role as a DNA binding domain. Adipocyte complement related protein, also called as ACRP30, which is of 30 kilodalton, was initially identified as a novel secreted protein highly expressed in adipocytes. Structural determination revealed close similarity previously unrecognized at the sequence level between ACRP adipocyte complement related protein 30 and tumor necrosis factor alpha. This unexpected result gave direction to subsequent efforts to determine the mechanism of action of this protein. Structural determination of E. coli YRDC protein shows the presence of a concave surface with positive electrostatic potential, which leads to experiment showing preferential RNA binding. Methanococcus janashi MJ0226 and Bacillus subtilis MAF proteins structure basically shows the presence of nucleotide binding folds. Further tests confirm that the MJ0226 hydrolyzes non-standard nucleotides. Structural genomics helps to deduce structural as well as evolutionary relationship. This became apparent with the first two protein structures being determined, sperm whale myoglobin and horse hemoglobin. They were recognized as homologs even at low resolution because their common ancestry was clear from the three-dimensional fold, although their sequences did not contain any recognizable similarity. Coming to lessons or outcomes inferred from structural genomic study. The one important lesson or the outcome from this study is it is possible to construct large scale facilities which can determine structure of hundreds of proteins per year. The structures which are generated from structural genomic studies are of high quality and they have an important impact on scientific research. The third important lesson that we inferred from the structural genomic study is that rapid deposition of data in public databases increases the impact and usefulness of the data, and technological development and its validation has played a critical role in structural genomics. International collaboration and cooperation advances this field and improves the data sharing. Last but not the least lesson that has been inferred from structural genomic study is determination of protein structures would be hinting at their biochemical, cellular, molecular functions, evolutionary relationship and also their medical importance. Coming to future prospects of structural genomics. Structural genomics offer great opportunity for value creation through the formation of new businesses. Funded initially by the venture capital community, several paths exist along which these businesses can develop. The first is predominantly service oriented models in which pharmaceutical companies outsource structural determination access to their internal capacity. This type of relationship is going to generate a revenue stream for the new company and that will allow the infrastructure to be built. Second important business which can develop is strategic alliances. Emerging companies may choose to build depth of expertise in the structural biology of specific protein classes or proteins in a specific therapeutic area and so forth. Pharmaceutical companies, they gain access to this expertise through strategic relationship in which research funding, milestone payments and royalties from eventual product sales are negotiated. Such relationships may extend to collaborative compound designing for companies that build capabilities in chemistry. Third future prospect or the business prospect of the structural genomics include database modeling. So, as technological advances deliver large number of novel structures in a systematic manner, an opportunity will exist to present the information gained in the form of subscription access databases. Database access will allow the use of information for functional genomics and target validation, as well as drug discovery and lead optimization. The cumulative data will also be of very high value to the development and refinement of homology modeling tools. As structural determination provides complete three-dimensional and functional view of genomic biology, various approaches will be utilized to identify selective small molecule ligands for proteins on a genomic scale. So, this can be defined as structural chemogenomics and will undoubtedly provide new opportunities for drug development. Technology development and integration from gene to structure process will also be a fertile area for commercial development. Major opportunities also exist within the agriculture industry, where genomic strategies are unearthing many new targets for pest control, and in the industrial chemical industry, where process improvements based upon structural enzymology will be increasingly utilized. There is also plenty of opportunities for filing patent applications covering inventions made during the process of structure determination and annotation and during the new processes and technological development. To summarize, structural genomics will revolutionize the molecular biology and biochemistry, making pervasive the use of three-dimensional protein structural information. Having the determined structures will enable the researchers to engage in high throughput inhibitor designing. Structural genomics will help in increasing the understanding of the design principles of the protein and may have an application to protein engineering. Structural genomics will reveal new functions, indicate molecular mechanism and explicate mutation by promising to offer a comprehensive library of experimental and computational models. In addition to this, structural genomics provide the platform for the detailed studies on the molecular interactions and dynamics and for the elucidation of large macromolecular complexes using X-ray crystallography and electron microscopy. Technological development and its validation also played a critical role in the success of the high throughput structural determination. Structural genomics have contributed largely by adding the novel structures to the database of structures in PDB, which is readily available and accessible to the broader communities. Thank you.